Okay, hello to all of you. So now we're going to discuss chapter number 11. And chapter number 11 is basically the first step of gene expression or protein expression, um, and it is the transcription. So in this process, basically what happens is from the DNA strand, RNA strand is made. And in this process, a very important enzyme is involved, which is called RNA polymerase. So let me tell you a little bit about this process in general, and that's what is written on this slide. So this is the process in which the information which is there in the DNA is first of all converted into RNA, and this involves important enzyme called RNA polymerase. An important thing to know about RNA polymerase is it also works in 5' to 3' direction. It does not require a primer like DNA polymerase. Okay, so this enzyme has a capability of initiating the process of formation of the RNA strand. So in this process, DNA is used as a template and RNA strand is made, and that RNA strand is also called a primary transcript, or it's also called a RNA, or it's also called RNA, mRNA. Sorry, it's also called messenger RNA. Okay, so this enzyme also works in 5' to 3' direction, and there are two strands in DNA. One of the strand is used as a template strand, okay, and the other strand is not used as a template. One of the strand is used as a template, and a RNA strand is made which is complementary to the DNA strand. Okay, so this is some of the general things about this process, and we will talk about more things as we move forward in this lecture. So this is the important. So first of all, we are going to talk about this process in prokaryotes, in the bacteria, and then we will be talking about this process in the eukaryotes. So you can compare how the this process of transcription is different in prokaryotic cells and the eukaryotic cells. So the RNA polymerase, which is involved in the E. coli, basically it has it's a multi-subunit structure, and these are the subunits in this enzyme. It has alpha, then this is omega, beta, beta prime, and this is the sigma. So when all these subunits are together, then this is called a holoenzyme. And when this enzyme lacks this sigma subunit, then it is called the core enzyme. And this is the molecular weight of this enzyme. So we are talking about the RNA polymerase, which is present in the prokaryotic cells. So as I said, there are two strands in DNA. So this is a process in which one of the strand of the DNA will be acting as a template, and on the basis of the sequence which is there in that template strand, an RNA strand will be made, and that strand will be made in 5' to 3' direction, involving the enzyme RNA polymerase, and we just saw the structure of the RNA polymerase. Okay, so the strand, the template strand, which serves as a template for RNA synthesis, it is also called antisense strand, or it's also called negative strand. And the other strand, which is a non-template strand, it is also called coding strand, or it's also called sense or the positive strand. Why it's called a coding strand? Because this strand is used as a template for making a RNA strand. This RNA strand is complementary to the template strand, and this coding strand is complementary to this coding strand. So the RNA strand is complementary to the template strand, and this coding strand is complementary to this template strand. So the RNA strand which is made has the same sequence as the coding strand, but the only difference is this coding strand is a DNA strand, and the RNA strand, or the mRNA or the transcript which is made, that is RNA. So coding strand and the mRNA, they have the same sequence, but the only difference is coding strand is DNA and the mRNA or the transcript is a RNA. So remember, going back to last lectures, in the RNA there is no thymine, there is uracil. So wherever in DNA there is T, in RNA it will be U. So basically the holoenzyme, which also has a sigma in it, it recognizes the DNA, and there is a special sequence on the DNA which is called the promoter region. So this enzyme actually first of all binds to the promoter region. Okay, so basically you can see this is overall your transcription. So this is the template strand, this is the coding strand. So this strand is the template for making the RNA transcript. The RNA transcript is made in 5 prime to 3 prime direction. So this is the way it is formed. So if this is the 5 prime to 3 prime direction of the mRNA, the direction of the template strand is going to be 3 prime to 5 prime. Okay. So here in this process, a hybrid of DNA and RNA will be made, and they will be antiparallel in nature. Okay. So this RNA transcript is made with the help of the RNA polymerase. And then the next step in gene expression is translation, which we are going to talk in the next lesson, in the next chapter, that how from mRNA the proteins are made. Okay, so basically, as we just talked about, the mRNA, the RNA polymerase is first of all going to recognize and bind to the promoter region. Okay, so basically in this process, first of all, a closed complex is formed, then a open complex is formed. So what is a closed complex and what's an open complex? So closed complex is complex that initially forms between RNA polymerase and DNA before transcription begins, and then this complex becomes open. So that's so this is a complex of RNA polymerase and DNA that occurs during the transcription. So in the initiation, so the transcription process involves 3 stages. First is initiation, then is elongation, and then the termination. So in the initiation, basically RNA polymerase binds to the promoter region of the DNA, and it binds to the DNA. Sorry, one second. Okay, yeah. So the information about the promoters is given here. And then the 2 strands of DNA, they are going to open up. Okay, and then the RNA polymerase actually moves on the DNA, and it keeps on making the RNA transcript. Okay, so only a specific part, something like around 17 base pairs, opens up. So the DNA strands, it's a double stranded, only this much part of the DNA opens up, and RNA polymerase actually moves on the DNA, and it keeps making the RNA transcript on the basis of the template strand. This is what I just said. And also there is the involvement of the enzyme called topoisomerase, which basically, as the DNA will be opening, there is a possibility that the DNA which is ahead can get very tightly supercoiled. So that's a function of this enzyme to open, to relax the DNA. And RNA polymerase also catalyzes the formation of phosphodiester bonds between the incorporated ribonucleotides. So this is basically showing how this process, and this is showing the initiation and the elongation. So you can see this DNA, this double stranded, this is the promoter region, this is the enzyme RNA polymerase with its sigma subunit. This is a complete enzyme. So it goes and binds here on the promoter region. Okay, so this is the initiation process, and then after that the 2 strands of DNA. So you can see how the 2 strands are together here, and when the enzyme binds here, the 2 strands of DNA, this starts opening up, this starts separating, and then there is a, actually in the promoter region there is a site which is called the start site. That is the first, so promoter is present on DNA. So there is a site on the promoter which is called start site. That is, so in the start site is the first nucleotide. So there is a nucleotide on the basis of which the first ribonucleotide will be made, and the formation of RNA transcript will start. So you can see how these DNA strands are separating, and how the formation of the ribonucleotides take place, or the formation of the transcript starts taking place. And then you can see how this this RNA polymerase, it actually moves on the DNA. It is actually moving on the DNA. As it is going to move on the DNA, first it was bound here on the promoter, now it has reached up to here, and as it is moving on the DNA, it is only opening a very small part of the DNA. And when the 2 strands they separate, one of the strand which is acting as a template strand, it is going to be read, and on the basis of the sequence in the template strand, the complementary ribonucleotides will be attached to this growing transcript. Okay, so and as the RNA polymerase has moved further, the two strands of DNA which are behind the RNA polymerase, they will wind again. So it is coming back to the normal. So only the part here of the DNA is opened up, and the template is going to be read, and the RNA transcript is being formed. So this is the initiation and the elongation of the transcription process in the prokaryotes. So termination, how does this process stops? It can take place by two processes. One is called intrinsic termination, and second is called row dependent termination. So intrinsic termination is basically controlled by some of the sequences which are there in the DNA, and the row dependent termination is actually dependent on the presence of a protein called row protein. So first of all, let's talk about the intrinsic termination. So you can see there are some regions in the DNA. These are inverted repeat regions. So this is the template strand. This lower strand is a template strand and is a coding strand. And so this is the mRNA. So this mRNA is going to have the same sequence as the coding strand. The only difference will be instead of T's, there will be U's. So this is the mRNA strand which is being formed, and you can see that because of these inverted regions, so they can, this is mRNA, they can actually form bonds with each other, and that results in the formation of the hairpin loops. So these are looped regions, we call them hairpin loops, and because of the presence of these looping which is taking place between the ribonucleotides in the RNA strand, that results in the weak binding between DNA and RNA, and that results in the termination of this process. So this is intrinsic termination, which is because of the some of the sequences which are present in the DNA. The other kind of termination takes place with the help of the protein called row protein, and it is explained here. So it's a protein. So this is shown here. This is a row protein. So this is the process of transcription taking place. You can see these are the two strands of DNA which have opened up here. This is the RNA polymerase. So it is moving on the DNA, and it is making this RNA. It's making this mRNA. Okay. So in the row dependent mechanism, so the row factor attaches to a recognition site on mRNA. So here it is binding here. So this is the mRNA. This is the row protein. It's binding here and moves it along behind the RNA polymerase. So here is your RNA polymerase, it's moving. So this is RNA polymerase, this is row protein, so it's binding here. When RNA polymerase pauses at the termination site, so this is the termination site which this RNA polymerase will recognize. So once that happens, the row factor unwinds the DNA RNA hybrid in the transcription bubble. So when RNA polymerase reaches this termination sequence in the DNA, at that point this row protein is going to go and unwind the DNA and the RNA hybrid, and that separates the DNA from the mRNA. So mRNA is formed. This is ready to go and start the process of translation, and DNA goes back to normal. So this mRNA which is formed in the prokaryotic organisms, it is ready for translation, but in the eukaryotes, which I'm going to talk very soon, there are some processings which takes place to this mRNA, and then this transcript or the mRNA which is formed, this is not a mature form of mRNA, it needs more processings, and then it is converted into a mature form of mRNA. Then only it leaves the nucleus and goes to the cytoplasm, and it is ready for the process of translation. So now we are going to talk about the transcription in eukaryotes. So there are some differences in the transcription in prokaryotes and eukaryotes. So the basic process is same. The most important difference is the involvement of some proteins which are called transcription factors. So transcription factors are not involved in the transcription in prokaryotes, but they are involved in the eukaryotes. So RNA polymerase is involved. So first of all, to the DNA, these transcription factors, they go and bind. So there's a promoter region. So these transcription factors, they go and bind to the promoter region in the DNA, and first of all transcription factors bind, and then they make RNA polymerase bind to the promoter region, and then the process of transcription starts. So this is a very important difference between the transcription in prokaryotes and the eukaryotes. And another important difference basically is the RNA polymerase. There are three different kinds of RNA polymerase in eukaryotes which are involved. RNA polymerase is basically involved in the formation of rRNA. RNA polymerase 2 is involved in the formation of mRNA, and RNA polymerase 3 is involved in the formation of transfer RNA. So this is basically showing the structure of the promoter in the eukaryotes. So this is the structure of the promoter in the eukaryotes. So they have here a initiator element. So here is the start site where the so on the basis of whatever the sequence is here, whatever the nucleotide is here, the um, first ribonucleotide will be added to the RNA transcript. So in the promoter of eukaryotes, there is a theta region. The theta region is basically rich in thymines and adenines, and then there are some upstream elements on this side, and then there are downstream elements on this side. So this is the structure of a promoter in the eukaryotes. So as I said, there is the involvement of these transcription factors, and here is a table which shows you the different transcription factors which are involved in the transcription in eukaryotes. So this is the name. So this is TF2D, TBP, then this is TF2A, TF2B. These are different transcription factors which are involved, and they basically help in the formation of a transcription initiation complex. So you can see how it takes place. So this is the DNA, this is the promoter region, the theta region is there. So first of all, this transcription factor TF2D is going to first of all bind on the promoter region. Okay, it binds here on the promoter region, and it's followed by other transcription factors also. So once these transcription factors bind on the promoter, then the polymerase 2, because polymerase 2 is involved in the formation of mRNA, then it is going to go and bind here, and that results in the formation of a pre-initiation complex, and then when this complex is formed, the DNA strands is separate, and the open complex is formed, and the phosphorylation takes place of the RNA polymerase 2. So there is a region which is called the CTD region. It is the carboxy terminal domain in the RNA polymerase 2. It gets phosphorylated. Okay, and then so this is the initiation complex, and here the two strands they separate, and then the RNA polymerase is going to move. It's just like the prokaryotes only. It's going to move, and the mRNA will be formed, and so this is all part of the elongation, and when the termination takes place, at that point this RNA polymerase, it's going to be dephosphorylated again, and this RNA polymerase after dephosphorylation is ready to start the process of transcription again. So this is main difference between the transcription in eukaryotes and the prokaryotes, that it involves some transcription factors. And so there are some proteins which are involved in the elongation process, and these are called elongation factors. And termination begins by stopping RNA polymerase, and there is a sequence in the eukaryotic consensus sequence. Means there's a sequence in the DNA of the eukaryotes which is AAU AAA. Basically this is a sequence for the there's a sequence in the DNA which is, this is basically shown the sequence in RNA. So this sequence is a signal for the process of transcription to stop. Yeah. And the last thing I would like to talk in this chapter is about the processing of the mRNA. As I said, in prokaryotes, when the mRNA is made, it is ready for translation, but it is not like that in the eukaryotes, because in eukaryotes, once the mRNA is made, it needs further processing. First of all, the two ends of the mRNA, 5' and 3' end, they need to be modified. So on the 5' end is added a cap, which is basically nothing but a modified form of guanine, which is attached on the 5' side, and on the 3' side is attached a poly A tail. So it's a long chain of adenines which is attached on the 3' side. So why the two ends of mRNA they are modified? Because mRNA is a RNA, and RNA is a very unstable molecule. This mRNA has to move from the nucleus and it has to go to the cytoplasm, and it has to go and bind to the ribosomes, because ribosomes are the protein factories. So the process of translation is going to take place on the ribosomes. So this mRNA has to go from nucleus to the cytoplasm, and on the way this mRNA is going to meet various enzymes, and these enzymes can degrade the mRNA. If the mRNA gets degraded on the way, then no protein will be made. So the two ends of the mRNA they are modified, the two ends of mRNA they are modified, so the mRNA is not degraded on the way. It's it's a way of making mRNA more stable so that it can be translated. And also another important thing to know in this processing is splicing. So in our DNA we have a lot of sequences in our DNA which we call junk DNA, which means those sequences do not code for the proteins, and they are called introns. Okay, and the part of the DNA which is coding for the proteins, they're called axons. So the third modification which takes place in the mRNA is, in the processing of mRNA, is these introns they are removed and these axons they are joined together. Okay, this is also part of the processing. So you can see this is the DNA which is going to be converted into mRNA. So these are the axons, these green ones are the introns. So these introns are junk DNA, they do not code for any protein. So when the from DNA, when the information is converted into mRNA by the process of transcription, so that information is also carried on, is also coded in the mRNA. So you can see the introns are there in the mRNA also. But before this mRNA can go to the ribosome, these all introns they are removed and all these axons they join together. This is called RNA splicing, and this results in the formation of a mature form of mRNA. So its two ends are modified, poly A tail, 5 prime cap here, and all the axons are joined. So this is important difference which does not take place in the prokaryotes. So this is what I just said, and this is showing you in more details. So this is a 5 prime splice side, this is a 3 prime splice side, this is in here a cut will be made and this axon will be separated, and then this side, then there will be a cut made here, this intron will be removed, and this axon and these axons they will be joined together. So this is all what I would wanted to tell you in the process of transcription, and in the next chapter we're going to talk about translation. And if you have any question, please let me know.